So I'm going to be explaining the SARS-CoV-2 life cycle. I realized in the past couple videos I did that the pictures were very small, so I'm going to have to kind of be outside of this frame and just to kind of explain with my hands. So that is, yes. So for starters, I thought I'd just explain the structure of the virus. So on the outside we have spike proteins or glycoproteins, and then it is enclosed in an envelope. Not every virus has an envelope, and not every virus has spike proteins, but this virus does. It has a capsid made of capsomeres, so that's what's shown in red. And then it has a positive sense single-stranded RNA genome that is dimeric, so that means there is two of them. So for the first step, I have attachment. So basically for this step, basically just the spike protein attaches to the ACE2 receptor on the outside of a human cell or other eukaryotic cell, because it can infect, like, I think dogs and cats. And basically it will attach, and it will not attach to prokaryotic cells because this virus does not want to infect prokaryotes, and it won't. So for step 2, I have entry. So this cell, instead of going through fusion, it goes through receptor-mediated endocytosis. So what this means is that the virus, once it attaches to the ACE2 receptor, it's going to kind of be engulfed by the cell and kind of fuse with that receptor, and once it is engulfed, it creates what's known as an endosome. So that is what happens here. For step 3, I have uncoding. So the endosome and the capsid, which it was the red pentagon shown, are disintegrated, and the RNA is released into the cytoplasm. And this is very essential because without this step, the RNA-dependent RNA polymerase, which we will be making in just a second, will not be able to get to the cell, and replication and transcription and translation and all of that will not be able to occur. So for step 4, it's quite unusual. Usually it's like replication or transcription, but for this virus, we actually go right into translation. I have the entire thing circled. It should just be a little part of the genome that is translated, and so it's going to use a host ribosome, and basically run along the 5' to 3' strand, because it can do that right away, because 5' to 3' is also what mRNA is, and mRNA is used to transcribe or translate proteins. And so basically this creates rDRP. I have a bunch of proteins, but it's going to create, for at least this one step, it's going to create rDRP, which we will need for step 5. Oh, and by the way, rDRP, I have it down here, is RNA-dependent RNA polymerase, meaning RNA-dependent, meaning that the template it is coming from is RNA, and the RNA polymerase is what it is making, so it's still making RNA. So for this step, we are going to use the RNA-dependent RNA polymerase that we just made in the cytoplasm, and we're going to run across one of the 5' to 3' strands of the genome and create a complementary 3' to 5' strand. And we can't use this type of negative sense RNA really for anything, because we can't use it for the genome and we can't use it for proteins. So we have to, our RNA-dependent RNA polymerase has to run across it again. It should be on the other side. And make a 5' to 3' strand. So, and basically that cycle just repeats in order to get a bunch of 5' to 3' strands for the genome as well as mRNA. So for step 6, I have translation again. So instead of making rDRP this time, we are going to be making proteins that we need in order to make the new viruses. So these ones I don't have circled because these I have used as the genome, and then these 3 I have used for protein making. So this still occurs in the cytoplasm. All this does, all of this occurs in the cytoplasm. The nucleus is there just to remind us that it infects eukaryotic cells that have nucleuses. So it's going to occur in the cytoplasm. A ribosome is going to run across it in a 5' to 3' direction. It's going to bind to a Shine-Dalgarno sequence, or a ribosomal binding site, and translate it to proteins. So this could be capsomeres, this could be spike proteins, glycoproteins, and basically proteins that are just needed for step 7, which is assembly. So for this one, we have our RNA genome that we just made in step 5, and then we have our proteins we just translated in step 6. And basically what I have here is everything is formed into a new virus, and the envelope that is for this virus is derived from the host cell. So that is what occurs here. So now you have new viruses. And so then the last step you need to do, or the virus needs, is budding or release. So basically this virus is just going to bud off of the cell. The envelope is going to kind of enclose it, and now it is ready to go infect other eukaryotic cells. So that's the end of the cycle. I just have a little note that the end result is usually cell death, also known as apoptosis, because it's such a complex cycle that basically at the end of it, the the host cell is just too exhausted to function. So that is the end of my SARS-CoV-2 life cycle.